Hi everyone, I am Jannat Shirin, Assistant Professor, PG Department of Microbiology from Econtent Development Center, Majlis Arts and Science College, Puramannur. Today, I am going to discuss about nitrogen cycle and symbiotic nitrogen fixation. Nitrogen is the constituent of amino acid, amino sugars and nucleic acid. In the atmosphere, nitrogen constitute about 78%. Valency state -3 to +5. Plants require nitrogen in large quantity, but it is not available to the plants because of the high stability of the nitrogen triple bond, that is N triple bond N. Plants and animals require nitrogen, but the ability to fix atmospheric nitrogen is restricted to some microorganisms, RK and symbiotic associations. Then the reservoirs of the nitrogen are soil organic matter, dead and living organic matter, humus and inorganic ions like ammonium, nitrate and nitrate. The photosynthetic activity of the plants could not take place without the availability of the fixed nitrogen provided by the microorganism or by the synthetic fertilizer. Next, we can move to the nitrogen cycle. There are mainly 3 stages in the nitrogen cycle. First one, ammonification. Second one, nitrification. Third one, denitrification. Ammonification. It is the conversion of atmospheric nitrogen into the ammonia. That is the ammonification. The amino acid produced by the protein degradation are further acted upon by the microorganism and produces ammonia. There are certain microorganisms involved in the ammonification like Micrococcus, Proteus, Pseudomonas, etc. Many plants, animals and microorganisms are able to carry out ammonification. There are mainly 2 stages in the ammonification. First one is proteolysis and second one amino acid degradation. Proteolysis is the breakdown of protein into simpler forms. That is, the protein is first hydrolyzed by the enzyme protease, which is then acted upon by the another enzyme called peptidase and forms amino acid. Okay, that is, protein is converted into peptide then to amino acid. Okay, some of the examples for the proteolytic bacteria are Micrococcus, Pseudomonas, Bacillus and Proteus. Second step is the amino acid degradation. The amino acid undergo degradation to form ammonia. Example, alanine plus half O2 gives pyruvic acid plus ammonia. Here the enzyme alanine deaminase catalyzes the reaction. Second step in the nitrogen cycle is the nitrification. In nitrification, ammonia or ammonium ions are oxidized to nitrate. That is the nitrification. Okay, there are 2 steps are involved in this process. First step, the conversion of ammonia to nitrite. This is an oxidation process and this step is carried out by the organisms like Nitroso-vibrio, Nitrosococcus, Nitrobacter, then Nitrosomonas and Nitrolobus. Second step is the conversion of nitrite to nitrate and this step is carried out by the organisms like Nitrobacter, Nitrospira, Nitrospina and Nitrococcus. Then the third step, that is the denitrification and nitrate reduction. Okay, it is the reduction of nitrate to nitrite and finally into the atmospheric nitrogen. Many heterotrophic bacteria are able to carry out denitrification. Example for the denitrifying bacteria are Thiobacillus denitrificans, Micrococcus denitrificans, Hyphomicrobium, Vibrio, etc. We can move to the next topic, that is fixation of molecular nitrogen. The atmospheric nitrogen made available to the plant is called nitrogen fixation. There are 3 types of nitrogen fixation, that are physical nitrogen fixation, chemical nitrogen fixation, and biological nitrogen fixation. Physical nitrogen fixation is also called atmospheric nitrogen fixation. Chemical nitrogen fixation means the conversion of atmospheric nitrogen into nitrogenous compounds by man-made chemical methods. Example, Haber-Bosch process, that is N2 plus 3H2 gives 2NH3. Here, high temperature and pressure are used to convert nitrogen into the ammonia. Okay. High pressure means 200 atm pressure and the high temperature 400 degrees Celsius is used. Then third type of nitrogen fixation is the biological nitrogen fixation. The conversion of atmospheric nitrogen into nitrogenous compound by microorganism is called biological nitrogen fixation. There are three ways of biological nitrogen fixation, that are symbiotic nitrogen fixation, non-symbiotic nitrogen fixation, and associative symbiotic nitrogen fixation. Non-symbiotic nitrogen fixation occurs in the free living organisms. Many bacteria and cyanobacteria are included in this group. Then the second type, associative symbiotic nitrogen fixation. This type of fixation occurs in the roots of grasses and cereal plants. Example for the associative nitrogen fixer is the Azospirillum. Next is the nitrogen fixing bacteria. There are many bacteria are able to carry out nitrogen fixation, that are Azospirillum, Azotobacter, Beechernica, Bacillus, Chromatium, Chlorobium, then Clostridium, then Rhizobium, Rhodomicrobium, Rhodospirillum, Desulfovibrio, Vibrio, Pseudomonas, and Thiobacillus. These are the organisms involved in the nitrogen fixation. Symbiotic nitrogen fixation is carried out by microorganism which causes a tumor-like growth in plant called nodule. Here the enzyme nitrogenase used to fix the atmospheric nitrogen. There are many bacteria are able to carry out symbiotic nitrogen fixation, that are Rhizobium, Azorhizobium, and Bradyrhizobium. Rhizobium is a gram-negative rod-shaped organism and it is a free-living heterotroph. It is a fast-growing organism. Okay. Then the symbiotic nitrogen fixation include the formation of nodule. There are three stages in the nodule formation. First one, recognition and attachment. Second one, penetration and travel. Third one, Bacteroid formation and maturation of nodule. The first stage in the nodule formation is the recognition and attachment. Legume plant root produces variety of metabolites, thus allows the rhizobium to grow in the rhizospheric region. Then the flavonoid signals produced by the plant can leads to the exchange of signals and allows the attachment of rhizobium to the legume plant root. Then Rhizobium bacterium produces a specific protein called RICA addition. It is a binding protein which can bind with the calcium complexes present on the root surfaces. Then the genes involved in the nodule formation is called noduling. Second step in the nodule formation is the penetration and travel. The polygalactouronase synthesized by the bacteria depolymerizes the plant cell wall and it allows the entry of the bacterium to the root tissue. Then after the penetration, the root hair curling occurs. The root hair curling caused by the node factor synthesized by the bacterium, and also indole acetic acid initiates the root curling. Indole acetic acid is a plant growth hormone. Then after the root curling, the bacteria enter into the root cell. So the plant induces to form a cellulose tube called infection thread. Then the rhizobium bacterium moves through the infection thread and later it moves into the underlying root cells and finally it reaches to the cytoplasm. The node factor synthesized by the rhizobium stimulate the root cell division and it causes the nodule formation. Third step in the nodule formation is the bacteroid formation and maturation of nodule. After releasing this bacterium from the infection thread into the host cell, it is converted into a swollen irregular shaped branched structure called bacteroid. Bacteroid is surrounded by a plant derived membrane, that is peribacteroid membrane. So the bacteroid with peribacteroid membrane is called symbiosome. Symbiosome is the site of nitrogen fixation. Leg hemoglobin. Symbiosome secrete a hormone which enable the production of leg hemoglobin. Leg hemoglobin is a unique protein which is similar to the hemoglobin found in animal tissue. Leg hemoglobin protects the nitrogenase enzyme from oxygen. Nitrogenase enzyme involved in the nitrogen fixation. Because of the presence of leg hemoglobin, the nodule appear as pink in color. Heme component of the leg hemoglobin is synthesized by rhizobium and globin comes from plant. Nitrogenase enzyme. It is the enzyme involved in the nitrogen fixation. This enzyme is able to convert atmospheric nitrogen into the ammonia. It has two coproteins. First one, dinitrogenase, that is FEMO protein. Dinitrogenase has 2,20,000 molecular weight, and second coprotein is the dinitrogenase reductase, that is FE protein. It is 64,000 molecular weight. One molecule of dinitrogenase complexes with one or two molecule of dinitrogenase reductase. The electron transfer is from dinitrogenase reductase, then dinitrogenase, then cofactor, and finally to the nitrogen. Here H2 is involved in this reaction, and reduced ferredoxin, cytochromes, coenzymes and ATP are also involved. That is N2 plus 6 electron gives 2NH3. So nitrogenase is an equilibrium mixture of FEMO protein and FE protein. This is about nitrogen fixation and nitrogen cycle. I hope you got an idea about nitrogen cycle and symbiotic nitrogen fixation. Thank you.